I am Irene Litvan. Today is September 24th, 2019. I am here with Glenda Halliday. We are at the 23rd International Parkinson's and Movement Disorder Society meeting, and I have the pleasure of interviewing Glenda, who I know from a long time.
That's true.
So Glenda, tell me a little bit about your education, and more importantly, why did you decide going into neuroscience?
Okay, so I did my schooling in Sydney. Well, I was born in Sydney, Australia, one of 4 children, and then my father, who was a mechanical engineer, got a job in remote Australia, north on the tablelands, as in the mining, and so I did my schooling in a small town of 6,000 people, and in my final year, 2 years, there was only 36 of us, and I was one of 2 that then decided to go on to university. So it was in a beautiful area in Australia, very remote, on the top of the tablelands, and it was sapphire mining. So my father was a person who did all the development of sapphire mining. And he, it's an area of Australia, it was the highest town in Australia, and it's in the subtropical rainforests. So it was actually an interesting place to grow up.
A beautiful place.
Yeah, it was. And as I said, it's a very small community, so it was ideal. But then I came back to Sydney to do university, because in Australia the universities are only in the capital cities. And I didn't really know what I wanted to do, so I enrolled in medical science and mathematics because I really liked maths. And so then I did very well at maths, and so at the end of my degree I still didn't know what I wanted to do, and I decided during the degree they forced the science students to actually do what they call social science, at least some terms, and so I had done some psychology in my degree. Of largely mathematics and medical sciences. And in psychology I became enamoured with reward pathways, and so for my honours thesis research I started to do comparative neuroscience of reward pathways. And then at the end of that I still didn't know what I wanted to do, so then I enrolled in medicine and a PhD at the same time, and really spent most of my time doing PhD work. And the PhD was on comparison, because the reward pathways are largely dopamine, and so then I became very interested in comparing the dopamine systems between humans and the animal models that we actually do. So my PhD, I have really highly cited papers, which probably people don't know about at this meeting, that are actually on the reward pathways for humans, because at that time they weren't described, and so in fact people didn't think that the system was the same. So I have a famous younger time of dopamine in reward systems. At the end of my PhD, I still didn't know what I wanted to do, and I hadn't finished my degree, but I decided I thought at that time I was pretty good at research and I was enjoying it, and I thought, well, I'll just do a postdoc and see. And I had written to all these laboratories overseas that I had been looking at in my thesis research, and thinking, I could go to New York or London or somewhere in Europe. Wrote all these letters, and 3 weeks before I sent the letters, the university, unbeknownst to me as a humble PhD student, the university went on a cost-saving mission in Australia and decided that if you didn't stamp your letters airmail, they would send them sea mail. So that all of my letters before I finished my PhD went off sea mail, and so the only response to the letters was from Australians. So I went,
no wonder.
I went to Adelaide for my PhD, for my postdoc, which actually turned out to be fortuitous. and started to work on Parkinson's disease. So I started the first brain bank on Parkinson's. They employed me because in Adelaide they had not worked in human brain at all before, and so I started working with the neuropathology in Adelaide in Parkinson's disease and set up the first donor program in Australia for Parkinson's disease research. And then 6 months later, when all my letters had gone, I got offers to go overseas, but by then I had already started my work in Australia. So after I finished my first postdoc, I came back to Sydney. And in Sydney at that time, it was the beginning of the Sydney multi-centre study for Parkinson's disease. Lovely. And so I was very fortunate to then start to work with an amazing amalgam of lots of different people. Like John Morris was the person that set it up. He was a wonderful man. I don't know if you know, but he loves birds and spiders. So, and in Australia that's amazing because they're actually amazing there. But and Maurice Healy, which is the neurologist that followed every patient for the whole time. I don't know how she did it. Wayne Reed, who was the neuropsychologist, again, had followed everybody for the entire time. So the quality is identical. And I was the person who set up the brain tissue programs and the questionnaires and made sure that everything happened. John retired about halfway through, and then I took over the last parts of that study. So, and we finished, the last person in that study died at 35 years out from the de novo. So it was a really interesting time, and I was probably very lucky that all of those serendipity things happened. So, yeah.
Well, I think it is an incredible way of growing and flourishing. Who was your role model? It seems like you were coming from a very small town and you end up flourishing in ways that probably were unpredicted for you.
Oh, yeah, definitely. I mean, my father still halfway through when I had real jobs, still asked me, how's university? Because they stayed in the country town while when I was they only moved away when they retired, actually. So I was very fortunate, I think, when I went to Adelaide. There was a person there, Laurie Geffen, who was a great international person working in psychiatry. So different to really neurology, but very interesting person who did a lot of work on dopamine systems, basically. He, in my postdoc, I went to a fantastic meeting in Israel with Laurie, it was the International Catecholamine meeting, which I don't think they have anymore. And I met Lisa Forno. She's a dynamo. I couldn't, such a small lady, and her. I actually met her sister, her twin sister, at that meeting, who was travelling with her, and Stanley Farn. And I don't know if you remember, but Susan Daniels.
Yes,
from the,
absolutely.
Yeah, I did a Google on her the other day, and do you know that the PubMed actually doesn't go, you can't search past 2002. So if you do Susan Daniels, most of her work, which was in the 90s, actually she doesn't come up. It's incredible. Anyway, Susan Daniels, who I think a lot of people probably will have forgotten.
Not me.
Yeah. So, I mean, Lisa Forno was old when I met her. But Susan Daniels was like a young up-and-coming neuropathologist.
Yes.
And I thought that was fantastic. And, of course, Andrew Lees gave some of the plenary lectures, which was just fabulous. So, I couldn't role model myself probably on Andrew Lees or Stanley Farn, but Susan Daniels particularly, but also Lisa Forno was just fantastic.
Yeah.
So
those are really good role models.
Yeah. So and in Australia, it's a small place, but a fantastic place to do research because it's a small population. There isn't there's no hierarchy, and you really do have to work together to do anything. And so building teams and actually working together towards large problems is actually a common thing that you do in Australia. So I think also I was just fortunate that in circumstances, basically. Yeah. Probably like lots of people. Yeah.
Well, you're very collaborative, you're very productive. I mean, your career has been absolutely incredible. And you have really very made a major impact in the field of Parkinson's, neuropathology, and neuroscience in general. Do you think that if you were to redo your life, you would do it differently in any other way?
I still somewhat regret not doing an overseas posting because I really haven't worked. It hasn't really, I guess, made a difference, but working at a distance with international colleagues, I think, would be different to working close with international colleagues. So from that perspective, I guess I would have probably tried maybe a little harder, but the circumstances didn't lend to it at the time. But that's probably my one regret, not actually interacting closer with international colleagues sooner. Do you know what I mean? Like, yeah, so...
Well, for not doing it, you've done pretty well, I would say.
Yeah. It's, I guess the things that have happened is that you can bring your research and another theme of research together, or you can use your resources to enhance somebody else's resources. But it would have also been nice to have actually got into other people's resources in a nice way. But anyway, yeah, it didn't happen.
There are not so many women that are as well known as you are. What do you think led you to become so well known, and what were the challenges that you feel you may have had as a woman to walk through your scientific career?
I think that I think I've been fortunate. I have been in a place and doing things in research that I think hasn't really been done by lots of people, so there hasn't been a competition by male or female. So doing brain neuropathology is something that's, and I've had the luxury of not doing it with a clinical role because I never registered medically. So I've actually only done academic neuropathology, which I think is actually pretty unheard of, actually. Mm-hmm. So because of that, and I guess because I just pursued my passion without really worrying about having money to live or what I was doing. Like I sort of have this very focused thing, I'd like to do X, and then that's actually what I go for. So I haven't seen, I don't think I've had barriers, but it's been a more unique way of doing what I do. I think I am well known mainly because I do try and understand the perspectives of many different types of scientists. So I have, if you talk to me neurologically, I understand and can respond in an appropriate way. I understand that because I've been in the clinics, you know, I understand that you can, what happens, how things happen. Same with neurosurgery, because I've been in the neurosurgical units. Same with neuropathology. And everyone brings same at the laboratory, same with people who just work on cell cultures, you know, the variety, people who just do DNA and genes. Everyone has a lexicon that's actually quite unique, and the way they see the research and the perspectives is actually quite different to each other. So it's a bit like multiple languages. And I think the reason why I'm well known is because I've taken time to interact with people to understand their levels and their questions so that we can answer some of the important things like, and that was probably again luck when I was in when I came back to Sydney and actually spent most of the time with that really quite amazing set of people who really had questions. The questions were quite different to when we started those studies, to the questions halfway through, to the questions at the end.
It always happens that.
And but everyone brought perspectives, and the discussions were deep and unique, and you actually understood what I must admit, neuropsychologists, I mean, you probably know this well, they have a different perspective again, and each neuropsychologist actually seems to have a unique perspective, but I think actually understanding those perspectives and everything. So I think that means that when I give talks, if people tell me who the audience is, I can make the talks relevant to the audience that I have. And so I think from that perspective, communication is actually why. So has it been a problem being a woman? I would say that in some funny way, for me, it's been an advantage. There's two advantages I had. I was a woman, I was a academic researcher, and I was in a remote part of the world. And if you have to have a variety of things on your committees, I'm a very good person to have. And so from that perspective as well, as if you have a modicum of contribution to make, and you can fulfil multiple aspects of a committee structure, then you actually I think that I was just in the right time at the right place. I think now I think it has been hard for women. I mean, in Australia, when I became professor of medicine, I was actually it was 1% of all professors in medical fields. And that hasn't changed that much, actually. It's about 3% now. There was no women neurosurgeons when I was in the neuropathology department at a large hospital at the time in Sydney, and we had neurosurgical registrars, and so I was there when the first two neurosurgical registrars that were women came through. And tried to make sure that they had a vision of being professor of neurosurgery. I'm not sure if they made it, but I think that, so I think I was just lucky that I had a position that I wanted that no one else actually wanted, and that I had ideas and research that people thought were of value at the time, and so that's what happened. And I think internationally, I fulfilled a lot of the things that were requisite at the time as well. So I then became better known internationally, because it is difficult from Australia to become well known. Yeah.
You were the one that made the effort. You were the one that went beyond and tried to communicate and tried to learn and tried to go and find what was important and work on it in significant ways. In fact, being 1% or 3% of the professors in Australia is still very impactful. So if you were to give advice to a young PhD or physician or movement disorder person, what is it that you would say that would be what they need to do or how they need to be?
Yeah. Well, I would actually say, and this might seem trite, but I would say dream big. Like when I first started in my first postdoc, I thought I will work on Parkinson's disease because really it's only the dopamine system that's affected. And like I should be able to figure that out within like because I'm already done my PhD on that. If I can't figure out what's wrong with that, then, you know, and if that's the only thing affected, which everyone told me it was, that would be an easy thing to do. So I think Dream big and don't be disappointed when you find out things that are different to what you expected, and have the curiosity and to follow those, and also to make sure that if you do have a viewpoint that you can change it, to make sure that you are continuously questioning the viewpoint so that you can move forward. But dream big. I mean, I think people, young people particularly, seem to me not to actually dream as big as they should, because even if you get halfway towards the big dream, you've made a lot of difference, and that's, I think, the key to actually thinking about what you want to do. I guess too, I've not thought about what I wanted to do in position or but in a either, I would think, in terms of a practice or a research question. And so I think too that not really making it about yourself personally actually also then means that you will make an impact, because you're actually trying to impact something else rather than just an impact for yourself. So those would be the two things that I would say. And I would say that the people I know that have actually become very successful all have those, all have not only dreamed big but actually succeeded in at least getting there. So, and also made impacts on many other things than themselves.
So where do you think science is going, or neuroscience is going? Where is Parkinson's 10 years, 20 years from now? How do you see it?
Right now, the next few years, I'm a little concerned, because I think that at the moment, in general, in neurodegenerative diseases, that people are following individual proteins. And so for Parkinson's, it's synuclein, and for some of the others, it's tauopathies. And I mean, I'll be very happy if that's successful in the short term. And I think probably it will be successful in making an impact on the inclusion formation. But my work and many other people's work would suggest that the inclusion formations don't fit the patterns of the degree of cell loss. And so I think we might be missing something. And I guess eventually, I think in 10 years it will become more obvious. And it is obvious in geriatrics now that there's multiple proteins. Even if you think you're working on Parkinson's over the age of 70 or 80, there's multiple proteins that are depositing. So if you concentrate on just one, whether or not that will be as impactful for that person over time is possibly doubtful, even at the moment. So I see the key things that will have to happen, and I think people will do this, is to actually really understand the mechanisms involved with that very early cell loss, which seems to me to actually be disconnected to the protein formation. And I would be very happy if I'm wrong, but I think that people will actually develop better things about that cell loss, and probably I think in 10 years' time, or 20 years definitely, we'll be looking at multi-targeted therapeutics rather than independently single-targeted therapeutics, which is where currently the world is.
I fully agree with that. Thank you so much. It's been wonderful talking to you. I know you as a friend, I know you as a colleague, and it's been really a pleasure to know you even better today.
Okay, well, thank you very much for doing this. I know that it's a very busy schedule for you. Hopefully next time I'll do for you.